All right. Hello, everyone. Today I'll present to you the project that I've been working on, which is about the computation of feasible compositions in microbial communities. Microbial communities can be used for a variety of purposes. For example, they are used in the food industry for the production of dairy products or fermented products. They are also used in wastewater treatment and in the production of biofuels. They are also present and important in different geochemical cycles, and they can be found in the human body as well, for example, in the microbiome in our skin or in our gut. And especially the microbiome in our gut has been linked to the inflammatory bowel disease, which is why in the medical field, there is interest to produce prophylactic or therapeutic tools to improve human health. Now, studying microbial communities comes with a few challenges, as they are very complex systems, and slight changes can have an effect on the community composition. And these changes include the medium, the pH and temperature, compound availability, and the growth rate of the community. And this is why we created a system to calculate all feasible compositions of a community under such various circumstances. Now, what do I mean with community compositions? In a community, there are various members of different organisms. In this case here, we have a 3-membered community where organism 1 has a much larger abundance than the other 2 members. So we could say that organism 1 has an abundance of 80%. In this community here, the abundance of all 3 organisms is the same. And in the one here, organism 1 is not present at all. So we can say that organism 1 has an abundance of 0%. And this aspect becomes interesting when you look at the output of such communities. Depending on which byproduct or product you're interested in, you may prefer one community composition over another. All right, so how do we build such a community? The first step is to read in the SBML models and to create the closed-off compartments for each member in the community. In our case, we have 3 members and therefore 3 closed-off compartments. Now we create a shared compartment that encompasses all of these sub-organisms. And if we now look at the reactions of these organisms, we can see that the members are still not properly interacting with one another. For example, metabolite 2 is consumed by organism 1 and is also consumed by organism 2, and it is produced by organism 3. But as it is right now, there is no chance of these members to interact with one another, and therefore we have to merge these duplicate metabolites into one. And this would look like this. And here, as we said before, organism 3 produces metabolite 2, and it is consumed by the other 2 members. And you can see much better already how the members interact with one another. Now, we also have to introduce a community biomass reaction, and this reaction regulates the growth rate of our community. And finally, if you remember the last slide, we have to take the community composition into account. If we have a very large abundance of organism 1, this would mean biologically that organism 1 has much more cells than organism 3. And therefore, the flux through organism 1 should be able to go higher than for the other members. And this can be done by relating the flux bounds of the reactions to the abundance of the member in the community. Okay, so we know how to build a community. Now we have to look at how we calculate the community compositions. The first step here is to create the age representation of the community. And this generates a multidimensional polytope, which is formed by the intersections of the constraints of the model. So, for example, here we have a house-like polytope, which has 7 hyperplanes. So we could say 7 constraints that form the polytope. We have 2 hyperplanes at the side, 2 at the front, 2 for the roof, and 1 at the bottom. And if we are only interested in certain dimensions of this house or of our polytope, we have to project it down onto the dimensions we are interested in. And this would look like this. So here in this example, we have 3 different methods, 3 options of projecting our 3-dimensional model onto a 2-dimensional space, depending on which dimensions we are interested in. In our case, we would project our multi-dimensional polytope onto the dimensions that contain information about the abundance of the members in our community. And this would be the composition space of the community. The final step is to enumerate the vertices of our composition space with MPLRS, which stands for Multiprocessing Lexicographic Reverse Search, and is an algorithm that creates a list of the vertices of your polytope. So our result is just a list of vertices. Now, if we look at the results of our example community here, I calculated the compositions for 6 different scenarios with different biomass rates ranging from 0 to 5. And the axes of this plot are the fractions of the organisms in the community. So a fraction of 0 here on the x-axis for organism 1 would mean that organism 1 has an abundance of 0%. If it is at 1, organism 1 would have an abundance of 100%. And you can see here that the larger the community biomass production gets, the smaller the feasible composition space becomes. And this is because the organisms, they have to start working together, and it gets much harder to create feasible compositions where such a high biomass production would work. So if we take a closer look here, we can see that the orange composition space has 4 vertices. And if we take a look at 2 of the vertices, A and B, we can calculate the flux through the community like usual with flux balance analysis. And this would look like this with a rather high abundance of organism 1. Point B, on contrast, has a very large abundance of organism 2 and 3. So, but what if you are not just interested in the vertices, but in a point within this community composition space, as for example, point C here? This can very easily be calculated as or by a linear composition of our vertices. So 0.5 times A plus 0.5 times B equals our point C here, which is a mixture of point A and B. Now I also calculated the composition space for a genome scale model that contains 3 submodels, which are involved in acetogenesis and methanogenesis, and are used for the biogas production, as the community itself can produce methane. And here as well, you can see that the growth rate has a very high effect on the community composition. Now, I want to briefly mention the PyCoMu tool, which is a Python package that can do all of this, what I just mentioned. It can assemble the community, and it helps very much in calculating the community composition space. It can use any format supported by CopaPy as an input file, and the resulting community model supports the SBML format, and therefore you can use it as you would use any other metabolic model. It is currently on GitHub. You can try it out yourself, play around with different metabolic models and see what happens. Thank you very much for your attention.